Dr. Jeff Guptill. He is our first presenter for the day, and he is an associate professor of neurology at Duke University School of Medicine and serves as director of the Duke Early Phase Clinical Research Unit, which is the Phase 1 and Proof of Concept Clinical Trial Unit at Duke. Dr. Guptill's research interests include the development of diagnostic and predictive biomarkers and conducting clinical trials of novel therapeutics for the treatment of inflammatory neuromuscular diseases such as myasthenia gravis. His goal is to understand why available treatments do not work for some patients and to identify targets for developing new therapies to help patients with these debilitating diseases. He leads the Neuroimmunology Biosample Network, a multi-state clinical research network, and biosample repository for rare neuromuscular disease, particularly MG. He is also a member of the executive committee for the NIH-funded Rare Disease Clinical Research Network for MG, where he chairs the National History Study and Career Enhancement Core. He currently serves as the chair of the Myasthenia Gravis Foundation of America Medical and Scientific Advisory Board. So thank you so much for being here with us. I will stop sharing and you can go ahead and present your slides. And thank you so much for being here.
Great, thanks, Nicole. Slides, Nicole?
I can.
Okay, perfect. All right, so thank you all for joining today. I just need to adjust my screen here. Thanks everyone for attending. My topic for today is to talk about some ongoing clinical research activities and highlight some of the, what I consider at least some of the major studies that are ongoing so that you as patients and caregivers understand what is going on in the field and also what to look out for on the horizon here as some of these treatments and clinical research studies impact the way we manage patients. So as I said, we'll talk about the current research regarding treatments and care management opportunities in myasthenia. And to do that, I'll first review the current state of treating myasthenia with the intent of just level setting so that we all understand, how MG is treated. You're probably pretty familiar with this, although there is some variability across the country and the world in which how MG is treated, but also to highlight some of the limitations of the treatments that we have right now, which will inform why these other studies are happening to try to address some of these limitations. And specifically, we'll talk about the PROMISE MG study, the group called MGNET, which is the NIH-funded rare disease network for myasthenia. And then we'll talk about current pharmaceutical industry clinical trials, which many of you are probably following and have some awareness of already. So first, let's talk about the state of MG treatment. So I'd like to highlight this particular graphic because it really illustrates how far we've come in terms of the treatment of myasthenia. So if we just want to focus on the open circles here, which is looking at, unfortunately, the mortality for myasthenia over approximately 100 years from 1900 to 1990, and how dramatically how dramatically, unfortunately, that has reduced over the years. And we can see that some of the steepest decline in the mortality from the disease wasn't actually from the treatments that we use today, but was actually for some of the supportive measures when people got really sick. So for instance, like patients that ended up on a ventilator that had crisis, being able to support their breathing, and if they got pneumonia from if they couldn't handle their secretions and swallowed saliva that got down in their lungs, which we call an aspiration pneumonia, being able to treat that with appropriate antibiotics has done a huge amount for improving the lifespan of patients with myasthenia, to the point where now it should be quite rare for patients to die from the disease, and we have much more effective ways of managing it. So, what this leads to now is, trying to get this number, which has improved even beyond this 1990 graphic, to 0. And then really, ultimately, the goal obviously is to eradicate the disease, which we're not there yet, obviously. So here's just an overview of the current treatment options that we use for myasthenia. Most patients, when they're first diagnosed, the first medicine that they will receive is a symptomatic treatment, the pyridostigmine or Mestinon, which tends to have a modest effect on the disease. And most patients will require some additional treatment beyond that. And then I've broken down these other categories into what I consider immunomodulatory, that change the reactivity of the immune system without causing widespread immune suppression. And then other medicines that I consider more immunosuppressive, that they suppress the response of the immune system in a more widespread fashion. And then finally, thymectomy, which is removal of the thymus gland for patients with acetylcholine receptor antibody disease. So immunomodulatory therapy, for a long time, it was really IVIG and therapeutic plasma exchange that were used for this, typically for the sickest patients, although IVIG is used in some centers to manage MG chronically. And you're all probably aware of the approval of eculizumab or Saliris, one of the first medicines that's been FDA approved for myasthenia for the patients, again, with acetylcholine receptor antibody myasthenia. And then in this column, the immunosuppressive medicines, many of you are probably familiar or are taking these medicines. And these are typically the most common medicines that patients will take outside of pyridostigmine. So steroids, which have been used for many decades and are effective, but come with some pretty significant side effects, particularly if used at high doses over a long time period. And then azathioprine and mycophenolate, which are azathioprine is also known as Imuran, and mycophenolate, which is Cellcept. And then there are some other medicines that are used, but not quite as frequently. And then I've kind of put rituximab in its own category. I think of it kind of straddles this boundary between immunomodulatory and suppressive. It operates by wiping out or removing a population of cells called B cells that we'll talk about in more detail as we move through the talk. So we have a lot of medicines, and many of the medicines are quite effective, but we do have some significant limitations. And understanding these limitations will give you a better understanding of what research is being done now and why. So one of the limitations is that we're uncertain which treatment or combination of treatments is best for a given patient. And I put the snowflake up in the upper right-hand corner because because myasthenia has been described as a snowflake disease. Each patient is different. And we currently have no way to predict the response to individual treatments. We typically choose these medicines based on a variety of factors about a given patient, their age, their other medical problems, for instance, but we don't have a way to know in advance for sure whether a patient is going to respond to a treatment that we give them. And despite these treatments, at least a third of patients will have residual symptoms or may be refractory to the standard therapies that we use. And finally, as I alluded to earlier, the current immunotherapies that we use to maintain the disease that people take on a daily basis, in general, have broad immunosuppressive effects that can have limiting side effects. And these immunosuppressive effects can open the door for potential infections. So with that background, let's talk about how we are trying to address these problems. So the first study I'm going to talk about is called PROMISE-MG. And PROMISE-MG is funded by PCORI, the Patient-Centered Outcomes Research Institute. And the principal investigators for this study are Dr. Narayamaswamy, who you will hear from later this afternoon, and also Dr. Donald Sanders from Duke. And so this study is a multi-center study where we're looking at the comparative effectiveness of treatments for myasthenia. So these are treatments that are commonly used in clinical practice today. So they're not really experimental therapies. They're ones that we give to our patients every day. And comparative effectiveness or comparative effectiveness research here is comparing the benefits and harms of alternative methods to prevent, diagnose, treat, and monitor clinical conditions such as myasthenia. And what makes this type of study unique is that it's done in a real-world setting. So rather than an industry clinical trial where a lot of factors about the population that's being studied and the medicine and who receives it is very tightly controlled, in this type of study where it's more real world in the sense that we're taking patients and observing what happens to them in the clinic without a lot of boundaries put on to who gets to participate in the study. And so almost all of this data is data that's being collected routinely as part of clinical care in the clinics. And there are 19 sites in the US and Canada that are participating. And there are two primary objectives, although there are a number of other objectives that will be addressed with this study as well. The primary objectives are to look at outcomes in patients with myasthenia who received azathioprine versus those who received mycophenolate. And so many of you know that azathioprine and mycophenolate are two very common treatments that are used for MG, various subgroups of MG as well. And although we think both will work, it isn't clear whether one may be a little bit better than the other, and the time frame in which people respond to those medicines. So one objective is to shed some light on that. The second is to look at different doses of treatments that patients are receiving. It's common to see quite a bit of variability in the doses that patients receive in various clinics. And there are some treatment guidelines, some recommendations, and Dr. Narayamaswamy will go over this in her talk, that these guidelines suggest doses to use when treating patients, but we see quite a bit of variability in patients that are and are not receiving those doses. So we want to look at, are these recommended doses actually important? Are we recommending the correct doses for patients? And so overall, this study is intended to provide some real-world evidence to help guide the management of patients with myasthenia for these specific medications. And so the eligibility for this particular study was you had to be at least 18 years old and you had to have autoimmune MG. It didn't matter whether you had what antibodies you had or not, as long as you had a diagnosis of MG. The real key factor for enrollment in the study was that it had to be new onset disease, so very early in the disease process before people got lots of treatments previously. So no thymectomies or other immunotherapies could be given. You could have received some Mestinon. And the enrollment of the study was 167 patients, about 62% were male and 38% female. And the average disease duration was 1.2 years. And you can see in the graph on the right, most of these patients, whether male or female, it was a later onset group of patients, so in the early to mid 60s. And this is actually pretty typical for the clinic population currently that we see, is that seems to be more of this later onset MG that we're seeing in the clinics. And you can see the breakdown of the patients based on race and ethnicity. So unfortunately, a lot of these patients are Caucasian or white, and we have very little representation of other groups. This is not unique to our particular study, and is something that has been observed not only in other MG studies, but in other studies in neurology and elsewhere as well. And that's something that clearly needs to be we need to work on to improve our representation of different patient groups in our studies. So at baseline, we also captured information about how severe their disease was rated at the time that they enrolled in the study, and also some other characteristics about any treatments and their antibody status. So in the graph on the left here, this shows the severity of their MG, and just based on their MGFA class. So this is the Myasthenia Gravis Foundation of America clinical classification. And so this goes from a 1 to a 5. The higher the number, the more severe the disease. So you can see that most of the patients that were enrolled in the study had mild symptoms that were either only ocular, so only drooping of the eyelids or double vision, or mild weakness of their extremities. And this is also what we would expect in our clinic population, particularly those presenting to the clinic versus those that presented to the hospital, where they would be more severe. Most of the patients had acetylcholine receptor autoantibodies, again, exactly what we would expect in our clinic, and a small population that had MuSK antibodies, which, as we know, are much rarer than finding the AChR binding antibodies. And then in terms of treatments, the most common treatments that were started at the initial visits, again, no surprise, lots of patients received pyridostigmine or mestinon, and lots of patients received steroids, and then a much smaller group received other treatments. So the data analysis for this study is ongoing, and the results should be available in the next 6 to 9 months. And as part of those results, we'll be trying to answer those questions, those primary objectives that I mentioned earlier in terms of the treatment with azathioprine versus Cellcept at 2 years and the dosing of those particular medicines. And as I mentioned, there will be multiple other analyses that will be done on this group because the patients receive many other types of treatments as well. Okay, now we'll move to the next topic, which is MGNET. And I mentioned earlier that this is an NIH-supported rare disease clinical research network for myasthenia. This is the first time that the NIH has supported a rare disease network for this disease, so a lot of people are very excited about this. The overall principal investigator for this is Henry Kaminsky, who's from George Washington. And the overall objectives are to enhance our readiness for clinical trials in myasthenia, and to do that through several different approaches, one of which is to study the natural history of patients with myasthenia, with a particular focus on subgroups that have been sort of neglected, I would say, in prior research studies. And we also want to work on improving the outcome measures that we use to monitor disease that can be used in clinical trials so that we can have better outcome measures that we can use to study, like, say, new therapies. To address this limitation about who's going to respond to treatments, we're trying to develop biomarkers, which are measurements that are taken from a patient that can help us to understand how a patient may react or respond to a particular therapy, and to that also may be used potentially to monitor patients' responses in the clinic. MGNET also has a goal to increase the number of junior investigators, so researchers who want to focus their careers on studying rare diseases and particularly MG, to train the next generation. And we also want to improve awareness of myasthenia, and this is one way of doing that through this session today. So I'm going to mention 3 core projects that MGNET is involved with. The first one is called Exploring Outcomes and Characteristics of Myasthenia Gravis. This is Explore MG. The second is the Biomarker Discovery Program. And the third is looking at the immune cells that are causing the problem in MG, causing the disease. So the first study is called Explore MG, and this is a natural history and biorepository study. And so the goal here is to enroll MG patients early in their disease course within a couple years, and then follow them for 24 months. And we're recording aspects of their disease, like their symptoms at the time that they started having problems, how they were diagnosed, and their treatments, and then how they responded, whether they needed any rescue therapies or had exacerbations. And the goal is to enroll 400 patients. We will also be collecting biosamples at these clinic visits when they come, and we will be using those biosamples for future research. And these are blood samples that we're using. A separate sub-study is called ADAPT MG, and this was actually not a planned study at the outset of this of MGNET, but arose as a response to the COVID pandemic. And so this is a study to validate outcome measures specifically for use in telemedicine for evaluating patients with myasthenia. So for Explore MG2, you can see the sites that are involved in this here. There's a coordinating center that coordinates the activities of the various groups at the Duke Clinical Research Institute. And currently we have 51 patients enrolled with, as I mentioned, a goal of 400. We got started late on this study, mainly because of the pandemic. Right as we were about to start, everything's shut down. So we weren't actually able to get off the ground with this until very early this year. So we're trying to play catch up. So we're hopeful that we'll make our initial goal of enrolling 400. We might be slightly below that, but we're doing our best to try to address that and get as many people enrolled as possible. And then the ADAPT tele-MG study. So many of you probably attended telemedicine visits over the past year and a half. And that was a challenge for a lot of people, for patients as well as for the clinicians. And so it wasn't really clear how we should evaluate these patients. There seemed to be some ways to evaluate them that transferred over from an in-person face-to-face visit to a telemedicine visit. So some things that we could assess by using like Zoom and other methods of video conferencing. And then there were some other ways of assessing patients that kind of arose spontaneously. And this study is really to see which seems to work best. What's the best way to evaluate these patients? And although this seems to, on the surface of it, be only applicable during the current pandemic, we also think that this research will be helpful for clinical trials in the future. So if any of you have participated in clinical trials in the past, you know that it's quite a big time commitment, and traveling to the site typically happens many times over the course of a study, and you're there for quite a while. In the future, we hope that studies will be able to reduce the burden on patients in that potential barrier to participation by allowing more virtual visits where we can evaluate patients and assess them, how they're doing, and get important measures of how the MG is responding to a given therapy through remote visits. So that should help reduce the burden on patients in the future. So we're trying to address that through at least the initial stages of this through this, this study. So the second project is the biomarker discovery project. And this, this aspect that I'm going to talk about now really gets to the question of why some patients respond to a given therapy and others don't. And so in this particular example, they're looking at patients' response to thymectomy. And so what was observed from the landmark study that was published in 2016 is that there was a distinct population of patients that responded very well to thymectomy, and then others that didn't seem to respond very much at all. And we want to try to understand why that is. And this is looking at the activities of daily living scale that you may be familiar with. And so the approach that's being used here is to look at blood samples from patients before and after thymectomy using a technology called metabolomics to look for characteristics in the blood that could predict who is going to respond to the thymectomy. This is a bit of a complicated study, but basically you take the blood and do the profiling of the blood. And within the blood, there's our different proteins and fats and other components, some of which are breakdown components from processes that are happening in the body. And you profile that and then analyze it and then look at whether or not any of these analytes or things measured in the blood have a relationship to the outcome of the patient over time. And typically there are multiple iterations of this where there's an exploratory phase where you come up with, okay, these seem like they work. And then you want to check a different population to see if it still holds true and refine it a little bit. And then you may try to validate it in a larger population to confirm that your profile, your predictive profile actually works. And so where this is right now is that they, from the MGTX study, that thymectomy study, they've identified 4 metabolites that can predict with a high degree of accuracy who is going to respond to thymectomy at 6 months. And you can see this is called a volcano plot that lists some of these. So this is a very complicated analysis, but this is very encouraging to this point. It is an early stage, and again, it needs to go through several steps of validation to know, to confirm that these metabolites will be predictive. So this helps us not only potentially select patients in the future for thymectomy, but also gives us some insight into what may be actually happening. So why patients, like what is happening when patients get a thymectomy? Why does the disease get better in some patients? What is the mechanism? And so that may also help us to, even develop new therapies that can have that effect without doing the thymectomy. Maybe there's a drug that could be delivered that can change the immune response in a similar way to having the thymus removed. So we will need some further studies to validate that. The next is this isolation of immune cells. And so here this study focuses on patients that have MuSK antibodies, and they're looking at taking the blood, again, from patients and looking specifically at the parts of the immune system that are causing the attack on the muscle. And so here they want to identify where the autoantibodies are binding to the protein and how the antibodies are actually causing disease. And so this is important because if we can understand this much better, we may be able to develop very targeted therapies that get right at the underlying problem without causing any off-target effects. So next I'm going to shift to some pharmaceutical industry trials. And I'm just going to start by mentioning a few trends that we're seeing in the clinical trial world, which I think are good trends. One is that the studies are starting to become more inclusive. So we have enrollment of patients with MuSK antibodies, for instance, and enrollment of patients with seronegative MG. And in particular, this group in the past has been not been able to participate in trials for the most part. So, and we need to have evidence-based treatments to manage those patients as well. So that's, I think, favorable. And also patients at different stages of their disease. So in a lot of cases, treatments were, or these trials were for patients who had basically failed every other therapy in order to get into it. Now, like a lot of these trials are trying to enroll patients who are earlier in their disease course before they've failed like a lot of prior medications. So this is an overview of what's happening. These are active trials. I'm not going to be able to cover all of these. I'm just going to focus on the left-hand side here. These are some studies that are furthest along in terms of the mechanism and number of studies that are being done. But there are others. There are other like chemical messenger studies that are in startup. This is another symptomatic treatment for MuSK-MG that's being studied as well. So let's focus on the left-hand column. And to do that, I just want to go over what is causing the weakness in myasthenia, and then that'll help you to understand why these particular classes of therapies are being investigated. So we think about if we want to move our hand, we have a signal that comes down from the brain in a nerve. And you can think of this as like an electrical wire. So the signal comes down the brain into the spinal cord, and then the signal comes out of the spinal cord in a nerve. And this is a motor nerve, which then contacts the muscle. And this is what it looks like in real life. So this is the motor nerve. These long gray cylinders are the muscle fibers. And you can see all these branches that go out to make contact to the muscle, and this is the neuromuscular junction, and this is where all the action is happening in myasthenia. And so if we blow that up, here's the end of the motor nerve, and there's release of this chemical acetylcholine, which then interacts with some proteins on the surface of the muscle, and if everything goes well, then the muscle contracts. And so in MG, we have these antibodies that are attacking the muscle at different points, either the acetylcholine receptor or you may be familiar with the MuSK antibodies as well. So understanding where the antibodies come from and what they're doing will help us to understand like how we can develop therapies to treat MG. And so here is another schematic of what is happening. So down here on the bottom is the muscle cell, and we have the acetylcholine receptors that are in the muscle cell. And the antibodies are produced by a subpopulation of B cells called plasma cells. And these B cells mature or differentiate into plasma cells, and these plasma cells produce billions of antibodies. And in myasthenia, these antibodies are attacking the muscle cell, most commonly in the form of the acetylcholine receptor antibodies. And the antibodies themselves can cause removal of the receptor from the surface, so they can't do their job and communicate with the muscle. And the antibodies can also activate a set of proteins called complement or membrane attack complex, which destroy the muscle cell. So with this background, you can see that there are potential points where we can try to interfere with the process. So one, we can try to attack the B cells and plasma cells to stop them from producing the antibodies. we can go a little bit downstream and try to remove the antibodies that are attacking the muscle and activating complement, or we can attack complement directly and try to prevent that from happening. And so these are the basic mechanisms of the drugs that are in development, of some of the major classes of drugs that are in development right now. So I'm just going to highlight very briefly what's happening with each of these. So for the B cell and plasma cells, the approach that's being taken is to try to selectively target those cells and remove them so they can't produce antibodies. And then if you can do that, then the antibody levels will drop, and then hopefully the muscle can recover and people will get stronger. And so currently there are at least 2 trials that are active in targeting these types of cells, and they're targeting different aspects of these B cells. And so these both of these drugs were developed initially for some or were tested in some other conditions prior to MG. So one for cancer patients that had excessive production of B cells, and then others for multiple sclerosis and systemic sclerosis, and then another disease called NMO, which all of these are autoimmune diseases. And so you can see that there are 2 trials active in MG with this specific approach. Now we'll talk about FcRn inhibition. And here we're targeting the antibodies themselves. And so the antibodies, they're produced by the B cells, and they float around in the bloodstream. And with the red blood cells and other white blood cells, and these little Y stick figures are the antibodies themselves. And so this is a little bit complicated slide, but to orient, this is the bloodstream is at the top, so where the antibodies are circulating. And this is a cell called an endothelial cell, and they play a major role in the lifespan of antibodies. So in normal conditions, the antibodies are taken up by the endothelial cells, and they bind to this receptor called FcRn. And if they bind to the receptor, they get transported back into the blood, and they can continue circulating through the body. So under most conditions, that's a good thing. You want the antibodies around to fight off infections. But in the case of MG, these antibodies get recycled, put back into the circulation, and they can go back to their job of attacking the muscle. So if we can interfere with this interaction, we might be able to selectively remove antibodies from the blood and reduce the activity of MG. So here there are multiple therapies being developed that are based on antibodies that selectively bind to the FcRn inhibitor and prevent binding of the AChR antibodies. And when this happens, instead of getting put back in the circulation, they get degraded and removed from the circulation. And so the effect that this has is very similar to plasma exchange, which also removes antibodies from the blood. And so there are numerous therapeutics that target this particular approach. The most advanced of these is by a company called Argenx, and the medicine is called efgartigimod. And so they've completed a phase 3 study that was successful, and we're expecting that they will be presenting their data to the FDA soon. And so we should hear a result as to whether or not this medicine will be approved for myasthenia relatively shortly. And you can see that there are several other drugs that are in development as well, using this same approach. And finally, we'll talk about complement. So here we're trying to prevent the activation of complement and the destruction that occurs on the muscle cell as a result of complement activation. And so this approach was really proven with the eculizumab studies, which is now called Saliris, and is the product that's on the market for generalized MG. This is just some data from those trials that showed that it worked on patient symptoms as well as on measures that the clinicians measured. And so we know that it's effective for patients with generalized MG and AChR antibodies. And so now there are additional approaches that are being studied for complement inhibition. So one is also by Alexion, the company that makes eculizumab. And so they have another version that, rather than having every 2-week infusions, which is the way Saliris is delivered right now, it would be every 8 weeks, which is a pretty major advantage in terms of the administration. And there are other drugs that are being developed. A phase 3 study of a medicine called Zilucoplan by the company UCB Biopharma. And there are other complement targeted therapies that are in various stages of development for MG as well. So to conclude, in my opinion, this is a very exciting time for MG research. There's a lot going on. We should have data available within the coming year about how to better manage patients with existing therapies. We have numerous ongoing clinical trials that are targeting different mechanisms of the disease specifically for myasthenia, including the removal of B and plasma cells, the inhibition of FcRn to lower antibodies, and to target complement related damage to the muscle. And we're expecting that there will be new treatments on the market for MG within the next 1 to 3 years, which will be great to give us new options for treating patients. With that, we will always be in a position where we need to get smarter and smarter about how to select patients for a given therapy, what's the best for them. And we need to improve our ability to make MG trials more inclusive for patients and easier to participate. So with that, I will conclude.
Thank you so much. All right, so we did get a question that came in. This person says they're newer to the MG community, so this may be a silly question, but there's no such thing as silly questions, right? Like we all know that. Are AChR antibodies found in healthy people without MG or similar disorders?
Yeah, that's actually not a silly question at all. That's actually a great question. So it is, it's thought that you do not see those antibodies in patients without the disease. And that's one of the reasons why those antibodies are such a good tool for us to diagnose the disease, is that they really shouldn't be there in healthy people. What is interesting is that you can find B cells that do recognize the acetylcholine receptor in the circulation, but they appear not to produce any antibodies, and that you can find those in healthy people. So it's kind of a paradox there, where you can see these B cells present in healthy patients, but they're not reactive. So, and there may be something about why these B cells, become reactive that is really a key for our understanding that we don't know yet.
Okay. Are drugs like Rituxan considered chemo? Is that along the thought process?
Yeah, so I don't know. That's kind of a tough one. I don't really look at it as chemo. I think of chemotherapy as a treatment for cancer specifically. Mm-hmm. Like all of these are, they're engineered treatments that are used to affect the disease. But I think the connotation of chemotherapy also it comes with it some association with significant side effects, right? Or some an understanding that there's going to be some off-target effects that are going to greatly impact the patient in a lot of cases. And I don't associate that with rituximab specifically. So I don't really look at it as a chemotherapy.
Okay. Okay.
Can I just jump in there for a quick?
Yeah, Pushpa, please.
Hi, Jeff.
Hey, yeah, go ahead, Pushpa. What do you think?
So, yeah, so I think that's a really important nuance, because the name chemotherapy almost makes one worry about it. When we talk to our patients, they sort of worry when we say, oh, it's a chemotherapy. And if they ask you, is it a chemotherapy, I'll get that question too. But the point is that rituximab is used for some cancers, lymphomas and other cancers. So does that mean it's chemotherapy? I think it really goes to our understanding of cancers, that just like myasthenia gravis and many other autoimmune diseases where our immune system is causing problems, the problem is that our immune system is overacting and we have to tamp down that immune system. We are learning more and more about the immune aspects of cancer, about the body's own response that causes problems with cancer, or how you can channelize the body's own immune response to treat the cancer. So because of that, there is going to be, and you'll see some of the medications that Dr. Guptill spoke about just now are also used in some kinds of cancers. And so that overlap is going to become more and more apparent over time as we understand the working of the immune system and channel the same drug to do different things, including in patients with cancer. So I don't know if that answers your question.
Yeah, thanks, Pushpa. Yeah, and I think that, as you alluded to, when patients learn that a particular treatment was developed to treat cancer, it elevates their concern or the anxiety potentially about the treatment itself. But I don't think that necessarily has to be the case, and you have to evaluate like each of the treatments on its own merit, and not just because it was developed initially for cancer.
Okay. What about pediatric patients? Are they being included in these studies?
Yeah, that's an excellent question. I meant to actually highlight that on one of my slides, and I apologize for that. So yeah, there is a clinical trial that's active right now for eculizumab in pediatric patients.
Perfect.
Yeah, this is an area that clearly needs some more research. Is how to manage pediatric patients with MG.
Mm-hmm.
It is a difficult problem because we think of MG as a rare disease. 60 to 70,000 patients in the United States. Of that group, the number that are kids, it's much smaller. So to really make any headway in that, there really needs to be a multi-center collaborative group that's working on this, and there are studies that are in development for that, like these natural history studies to understand how we should be treating pediatric patients.
Good. Very good. Okay.
That's a great question.
Do levels of antibodies that are present in the blood but below the threshold of being diagnostically positive hold any importance?
Yeah, so that's another. These are great questions. You guys are good.
We're keeping you on your toes this morning.
So this is a common referral for us, is that, and for Dr. Narayamaswamy can attest to this as well, is that we commonly get patients who are referred to us because they may have autoantibodies detected in the blood, but they're not considered above the threshold for abnormal, or they're just barely abnormal. So it's a sort of a complicated scenario. Some of it depends on which lab is doing the test, and they can report the results of the test in different ways, where sometimes it may be listed as indeterminate, for instance, and that is typically problematic because it leads to like a lot of questioning about it, whether it's not positive or negative. It's kind of in the middle. It's in this borderline range. And in most of those cases, the indeterminate ones, we don't think that it is associated with myasthenia. And typically what we'll do is we'll do further investigation to try to confirm whether or not there appears to be any activity. And we typically will use neurophysiology tests like repetitive nerve stimulation and single fiber EMG to look at the nerve to muscle communication more directly to see if there's any abnormality there.
Okay.
Typically, we will not put people on medication for an indeterminate or subthreshold lab value and just watch them.
Okay.
If on the other hand it's indeterminate, but we can clearly see evidence of abnormal nerve to muscle communication on a single fiber EMG test, then we may treat them. But typically we would just follow them.
Okay.
The second part to this question is there are different aspects, different acetylcholine receptor antibodies that you can detect. So there's one called a binding antibody, and then some labs will test others called modulating antibodies and blocking antibodies and striated muscle antibodies. And all of those, except for the binding antibody, have less diagnostic significance. The one that we really pay attention to is the binding antibody. If that is positive, it is very highly associated with the disease. If someone just has an isolated modulating antibody that's a little bit above abnormal, that's again someone that we would investigate a lot more thoroughly to see if we can find evidence of abnormal nerve to muscle communication. But oftentimes we're just watching them and maybe have them come back in a little bit and retest their antibodies to see if the binding antibodies come back positive.
Okay.
That's a great question.
How do these newer treatments impact general ability to fight infection?
Yeah, these are great. So one of the reasons that a lot of people in the field are very excited about the developments of the clinical trials that are happening right now is that these approaches tend to be much more targeted than the existing treatments. So like prednisone not only affects the immune system, and within the immune system causes broad range of effects on the immune system, but it also has off-target effects in impacting blood sugar and bone health, et cetera, et cetera. Whereas these new treatments, particularly monoclonal antibodies, tend to be very specific and target a very specific mechanism.
Okay.
So the promise that they hold is not only in the therapeutic effect that we're trying to achieve, but also the concept that they should have a better safety profile. So not cause as widespread immune suppression and not cause as much concern about getting infections.
Okay. Do you know if any Native American or other indigenous people are included in these tests?
Yeah, so I think the approach is that everyone is, if you meet the eligibility criteria for the trial, you should be able to participate. And there are no eligibility criteria that would, exclude you based on race or ethnicity. That's not part of the trials. I think the real challenge is to, in a lot of cases, is the outreach and the awareness that these trials are available in different communities. So a lot of the trials are focused at centers that see large groups of MG patients, which typically tend to be in cities. And being able to have an effective outreach to other areas, rural areas and other groups is a real challenge, but could be a real source of new involvement or engagement for patients in clinical trials if we can reach those groups of people effectively.
Okay. And I want to be cognizant of our time, and we have quite a few questions, but I know that Pushpa can answer these too, part of her discussion. So let me just finish by asking, do any of these ongoing trials that you spoke about include any seronegative patients?
Yes. So several of the trials do.
Okay.
I don't I don't necessarily want to mention specific
Okay.
specific ones because I'll inevitably leave people out
Sure.
that
You're
aren't
okay.
including seronegative patients. But yes, yeah, and I think that's been a conscious effort on behalf of the pharmaceutical industries, as well as some of the people who are some of the researchers who advise the pharmaceutical industries, that we really want the seronegative patients involved in the clinical trials. Because we, as I stated earlier, we really need to have evidence-based treatments to help that population as well.
Thank you so much for being with us today.
Great, thank you.